Buenos días a todos.
Good morning, everybody. We're going to begin today's session. The first session has a title, Challenges and Opportunities in the Development of Advanced Therapies for the CTNNB1 Syndrome. And the first speaker will be Dr. Ana Gonzalez, who's Vice President of the CTNNNB1 Syndrome. Thank you.
It's on the second day, a very exciting one. I'm going to introduce you to the topics of today, show a roadmap of the molecular approaches and what we do at the foundation and why we do it. So first of all, what are our goals as a foundation? That is to tackle the disease from different angles with innovative therapeutic approaches. More than one, you will see. We would like to develop therapies, different therapies for different needs. Why? Because our children have different severity of symptoms. They have also different ages. And there is also a difference in accessibility in different countries and regions. We are also aware of that. Most importantly, we are ambitious for the future of our children, and we are open to explore other therapeutic options. We would like to improve the lives of all the CTNNB1 children. How do we get to do this? Well, we cannot do this alone. We do it thanks to our research network that is comprised by a top team of experts from all around the world that are helping us develop these different strategies. I would like to take a moment to thank all of them for the commitment they have with our project and with our children, because without them we will not be here today. Now, many of you heard of gene therapy, are looking forward that gene therapy comes in, but I'm not sure if everyone knows exactly what is gene therapy, how does it work. So gene therapy is a set of techniques that uses either replacement, inactivation, or introduction of genetic material to prevent or to cure genetic diseases. We have some very good examples that have been successful, such as child Parkinson's disease, ADCC, and also spinal muscular atrophy, SMA. There are some obstacles, of course, to these kind of therapies. There is difficulty in targeting the genetic material specifically to the cells of the tissues that you need. You also have to identify them first, which is not always easy. There is also the regulation of the genes. How do we avoid that we overexpress it or underexpress it? Because it could have detrimental effect on our children. Just to give you an idea, a very exciting overview of the gene therapies that have been approved in Europe in the last years. Although many of them got orphan designation a bit before, everything that you see there got market authorization in the last 5 years. The field is booming, and we are getting great examples that we can use to move forward. It's happening in the last years, and we will try to make it happen as well. Maybe for those who don't come from Europe, I also want to indicate which ones, for example, are available in the United States. Like you see there, I have made 2 columns. Those are 2 types of gene therapy approaches that we are going to follow. By the way, I switched them completely last night at 2 a.m. So the first row is actually GRT and the second is ASO. Sorry for that. We have examples like spinal muscular atrophy, like I said, child Parkinson, retinal dystrophies, and just like 2 weeks ago, market authorization was given to Hemgenix for hemophilia. Now, what are the current CTNNB1 therapy strategies that the foundation is approaching? First of all, we have the gene replacement therapy. That is introducing a normal copy of a mutated gene into the target cells, the brain, without it being integrated in the genetic material of the host. I think this is a question that many of you wonder how it works, so we are not introducing it with this technique in the genetic material of the children. This is being developed by the Children's Medical Research Institute in Australia, and you will hear their talks later today. Then from the National Institute of Chemistry in Slovenia, we have 2 approaches, 2 strategies. First, the antisense oligonucleotides, the ASO, and then the CRISPR-Cas. The ASO works at increasing the production of beta-catenin from the healthy allele, we are not targeting the one with the defect, but the other one. We're trying to increase the production of that one. And with CRISPR-Cas, what we are trying to achieve is through gene editing, cut and paste, to replace the mutated gene or part of it with a non-defective one. So to try to copy, paste, cut and paste and fix it. You will also hear really interesting talks today. I want to go a bit more in depth into the different strategies, but not too much. Also to show a bit the differences. The first strategy, gene replacement strategy, is a one-time solution. Once it's in, we cannot get it out. We cannot redose in short time for sure, or the science has not advanced yet enough to know if redosing could have a problem because of the immune system. And it is a type of therapy that, like we say, a functional copy of the gene will be inserted in our children. Then for the second strategy, that's something that you can dose. You can also dose at different time points. You can increase and lower the dose. It can be more adapted also to the weight of the child as they grow. But of course, depending on the results, sometimes one can be more effective than the other. And then for the DNA modification techniques, yes, that is the one that we said is copy-paste. And I don't have much information on the dosages for this one, so you can ask those questions to Dusko later. What is most important, I think, for most of you is to know that the way we envision this is that more severe phenotypes from the children might require strategies 1 and 3. They are also a bit more risky in the application, but we will do anything to control that. If children are less affected, maybe by the syndrome, they could be good candidates, for example, for the strategy 2, which is the one that you can dose at different ages. I'm going to focus very quickly on gene replacement therapy because that is, from our projects, the one that is more advanced and the one that you will also hear a lot of today, the one everyone is actually, I think, wondering about. So just to give you a very quick introduction, we take an adeno-associated virus, so it's a virus that is non-pathogenic, and our team is going to put a copy of CTNNB1 gene that is healthy into it. And you know, viruses replicate inside of us, so that's a bit, in short, the goal of it. The CTNNB1 gene, luckily, is a good candidate for this because it's caused by a loss of function mutation, so you can gain the function with this type of methodology or therapy. And also, the size of our gene is small enough to fit. This technique has limitations in the size, and we are lucky enough that we can try it. Now, our team, and they will show you later, has created a number of candidates with different combinations. And then what they are doing, what they have done is test it in cells derived from our children. The model that Dr. Akelan, for example, explained yesterday is the same system. And they are looking at the gene transfer, like, is the transfer of the virus optimal into the cells? Then, is it expressing enough? Is it working inside the cell, even in a model? And finally, how much protein are we getting from that? Do we see an increase? You have to test this before to see if your model is valid. Once this is done, we have moved on into mouse. You also will see this talk today. There, we check for other things, like when we treat a mouse, can we see a difference with non-treated mice? Like we will check for behavioral and cognitive tests. We will check for the efficacy of transduction. We will look at the tissues inside the animals, and can we find an increase of CTNNB1? Can we locate even the vector that we have introduced in them? And that's a bit how it works. If we, through this method, can find a suitable candidate that proves that it could work, we might be in the place to start moving into the clinical phase, which is also a long one, and you will see also a presentation on how this develops. Finally, I would like to show a quick roadmap of how we plan to get from detection into correction. First, the research that is being done at the Children's Medical Research Institute. There, they have designed the vectors, and based on the available data and the results, there is a selection being made. They go into the different models, cell models and animal models. These preclinical studies are being done also at the Children's Medical Research Institute. And then we will go into vector manufacturing. We have to multiply the candidate only if the candidate is successful. The timelines depend highly on the results we will get in mice, and they can be shifted. Only if that works, and only if the manufacture is optimal, which they can test for us, we might go into first in human, and eventually, we hope, into correction. With this, I would like to thank you all our scientists working on this, and introduce the next speaker into the stage. Thank you.